Good evening, everyone. I'm Shariq Mohammed. I'm an assistant professor in the Department of Biostatistics at the School of Public Health. I'm also a junior faculty fellow at the Rafiq Hariri Institute for Computing, Computational Science and Engineering. My work is mostly focused on developing statistical methods for various types of complex health data. So today I'll be focusing on some of the major sort of the broad overview of my interests in terms of my past work, as well as present three specific examples of projects that I'm currently working on. So my primary interests are working with complex data. One of the major examples is working with imaging data in the context of different disease applications. What you see here on the slides are imaging data in the context of gliomas. So in gliomas, it's well studied that tumor heterogeneity is associated with genetic mutations and survival. So I was developing statistical models that capture this heterogeneity in a structured way and use that quantification of heterogeneity to predict clinical outcomes of interest, as well as use this quantification based on imaging data to identify associations with genomic markers, because imaging-based phenotypes could potentially serve as non-invasive diagnostic tools. So to do this, my methods essentially are broadly focused on using Bayesian models that can incorporate structure of the data and give us uncertainty quantification about our inferences, using some variable selection priors, as well as incorporating functional data analysis within these methods to be able to understand the heterogeneity from these imaging data. Some of the current sort of projects that I'm interested in or working on include spatial single cell data sets. So we have these high resolution spatial transcriptomics data sets that give us access to exact spatial locations as well as gene expression at those locations. So can we leverage statistical methods like spatial statistics and modeling to understand and gain localized quantitative insights about these tissues or tumors in different disease contexts. So I'm using tools from differential geometry to understand the expression of different genes over this tissue surface and try to identify specific regions of interest in a quantitative and an automated way. I'm also interested in looking at understanding the interaction of different cell types within a tissue in a given disease application. Can we quantify interaction of different cell types, how different cell types mix with each other in this tissue space, and use that quantification to predict response to treatment, for example, in cancer or other diseases. Another example that I'm also currently working on is using digital neuropsychological tests as a potentially non-invasive early diagnostic tool in the context of aging and Alzheimer's disease and dementia. The two main tests that I'm sort of currently working on are called the digital trail making and the clock drawing tests. These tests are essentially you connect dots in a specific sequence as instructed, or you draw a clock with a specific time. These tests used to be administered with a pen and a paper, where you would only get very crude measures, such as how many errors did you make, how long did you take to complete these tests. But with the digital neuropsychological test, you actually get high frequency pen movements that are recorded at every 13 milliseconds. So we're trying to leverage these high frequency, high resolution data, trying to capture as much information as possible to not only generate these crude measures that were possible earlier from a paper or pen-based test, but also come up with new new measures that could potentially predict progression or even distinguish between different categories of dementia. Another project that I'm also involved in is something that Kevin talked about earlier in the session, where we're working with industry partner ShareCare, where we have access to individual level responses or individual level survey data about health and well-being related measures. And what we're trying to do is leverage those individual level information to quantify how zip codes perform in the context of health and well-being. And while doing this, we're trying to bring spatial proximity into the modeling framework, meaning if two zip codes are neighbors to each other, if two zip codes are within a certain proximity of driving distance or within 30 minutes to each other, for example, we try to incorporate that type of neighborhood information to capture similarity between these spatial objects and use that to summarize information, individual level information, to a zip code level analysis to sort of understand how these zip codes might be performing on various metrics of health and well-being. If any questions or any interest, happy to talk about it. Thank you.
Just click the mouse.